But thank you very much for having me here to talk about science, the day job. So I thought these two images kind of summed it up for me. Exciting and oh dear God, what am I doing? So the main topics I'm going to cover is immortal cell lines, protein lysis and quantification, separation by SDS page, immunoblotting, chemiluminescence, and it's way less complicated than it sounds. But the first thing I really want to talk about are proteins. So that's really what I look at every day, is proteins. Like Claire's going to talk to you, I think, about DNA, I hope. And I'm basically, what I really look at is the prion protein. That's what my group studies. And proteins obviously are made up of 20 amino acids. And the ones you probably know, like casein and lactose in milk, or cellulose in plants, keratin in your hair and nails. Alcohol dehydrogenase, extremely important. And these are assembled from amino acids using information encoded in our genes. And obviously their structure is important for their function. So you can kind of see over here, like here's the primary structure, which is the way the amino acids are put together. The secondary structure is really these two types of structure, like alpha helicase and beta sheets. And then your tertiary structure is how they start to fold in together again with hydrophobic or hydrophilic interactions. And then obviously there's this other quaternary structure, which actually becomes when we start looking at protein complexes. So we'll keep it simple. Like many proteins carry out obviously essential processes in the body, like thrombin converts fibrinogen to fibrin, which allows clotting to happen, and that's pretty important, I've heard. So the main sort of thing is the first thing you want to look at when you're doing my kind of work is what's your protein sample? What are you going to take proteins from? What proteins you want to look at, and where do you want to find it? So there's sort of a few ways to go about any project, and it sort of really starts. You want to look at in vivo samples, so human, animal samples from real life samples. So Alyssa is going to talk about using patient samples, but there's some problems with getting these. There's not exactly like loads of people running up to try and give you samples of themselves to test in labs. There's sort of a rarity. It's quite a privilege to get to work with patient samples or human animal samples. A lot of clearance, a lot of ethics. You don't want to take loads of rats or mice or animals or monkeys or something to be used when you don't really have a clue what's going on. Very unlikely to kind of just give you animals or human samples to just chuck some stuff at and see what sticks. You have to have a really good sort of a sound basis. And then also, if you don't have enough samples, can you draw like relevant data from it? So the conclusion, but sort of the start to start things off, you can really go with in vitro samples. So immortal cell lines are used quite frequently, but then you've got to sort of say, these are cell lines that are dividing in a dish, or these are things that are sort of taken out of the human system or animal system. Like, are they going to actually be reflective of reality? Is what you're going to see when you put things in a dish in an incubator going to be what is actually happening in a human body? Because obviously those conditions, I've heard, are slightly different. And then you've got, like, if you wanted to go down the roads of synthesized peptides, people are looking at like more synthetic, lab created things as well. And then again, you have the problem of, will that be in any way reflective of reality if you've just made it yourself? So I work on immortal cell lines. So these are cells that due to a mutation can grow indefinitely. So there's a lot of the original ones would have been taken from cancerous biopsies. So for example, the first immortal cell line are HeLa cells, and these were derived from a biopsy of a particularly aggressive cervical cancer that Henrietta Lacks had in 1951. And I'm just going to quickly say that Henrietta Lacks didn't actually give any sort of approval to have her biopsy used for research or anything like that. And so there has been sort of some ethical concerns because her family then found out and actually didn't want people to be using it. They sort of felt like having those cells there would still mean that she was alive in part. But these cells actually did go on to sort of have some massive breakthroughs in cancer and things like that. There's a lot of sort of background on that, and they're even considered sometimes a bit of a pest in immortal cell culture because they're so good at growing that they can actually just jump into other dishes and sort of start infecting your other cells, and you basically will have what you thought was one immortal cell line and then ends up being a load of HeLa cells because you've accidentally let one cell in and that's taken over. But immortal cell lines are invaluable tools for experimentation, I think, because you can kind of replicate what as much as far as you can in vitro replicate what's going on without using multiple tissue samples and things like that. So I think it's all very, it sounds very cool, but what's the practicality? What's it like actually working with these? And it's really like looking after a precocious pet. They don't really care that it's Sunday or the weekend or a bank holiday or that you've been very careful or that you were tired. If you get any sort of infection in them or bacteria, they are unforgiving and you will have to start again. So this is sort of like a plate, so I'm just going to show you sort of from this oncology lab. This is sort of like, this is exactly what I would have. This is like a 6-cell dish, I think you can see. And you can see there's like a pink media in there. Now that media will have all the nutrients that you need for the cells. Obviously that's completely sterile. You don't want any bacteria in it at all. It's made up with fecal calf serum and then different essential amino acids and sugars and stuff. And the color as well will change. So pH is really important for growing cells. So the cells give out, obviously as they're growing, give off different gases. Carbon dioxide, and that will change the pH of the media. As the pH becomes more acidic, the media will change from pink to yellow, and it's a good indication that you need to change the media. Sometimes it doesn't often go like that either, so you need to just really monitor the cells, sort of know how they're going to grow, and look after them. So what she's doing is now she's putting them in a fume hood, or not a fume hood, a laminar flow cabin. So this is a sterile environment. The air is being sort of pushed out and displaced, and you have to do everything sterilely with cells like these. Get anything in them, it's going to completely take over, especially if you don't have any antibiotics. And you just have to be really careful. So that's that. Oh yeah, and I'm going to just show you, when you look down on a microscope, what these cells, what cancer cells, or what maybe your cell culture will look like. So these are normal cells, and you can see, I don't know if you can see, it's not the best video here, but you can see they're kind of dividing kind of slowly, they're just growing kind of normally. I can actually speed this up. And this is sort of what a normal, like if you had non-sort of cancerous cell culture, that's what it goes. Now that would be very difficult to use because over time you just wouldn't generate enough cells. But you can see this is a cancer cell, so this is the sort of things we would use. In 3 days I can split 1 dish of cells into 5 dishes of cells, and I'll have 5 dishes of cells. So you can see these divide crazy and they just take over. Some cancer cells have sort of cell migration as well, so, and I'm not going to show that because I just showed it. Yeah. But, so yeah, so then you've got your samples. So you've got your immortal cell line or whatever, you maybe treated them with drugs, you've got your brain's neuroblastoma cells that I work with. I want to look at the proteins in them though. So first I actually need to bash open the cell wall. So we just use a mixture of detergents, or you can use usually SDS or NP40 and things like that. Different detergents literally just punch holes in the cell wall, release all the proteins. And then you'll put some inhibitors in there to stop all the proteins being broken down, because obviously in our body and in cells they need to control the type of proteins that are going on, so they have loads of proteinases that break down all the different proteins. So you just lyse open the cells using a detergent. It's not really that complicated. So then we have a soup of all different proteins, all the proteins that are in our cell. How do we look at what we're interested in? Maybe we're only interested in a particular protein. So the first way to start going about that is to separate the proteins based on size. So for example, like actin is quite big, which is makes up our cell walls, and that's about 42 kilodaltons, which is 42 kind of mid-range. And then for example, superoxide dismutase, which is an oxidation protein, that's about 7 kilodaltons, so it's pretty small. So what you're going to do is you first take SDS and you coat all the proteins in a negative charge, and then you make a jelly mix of polyacrylamide, and you're going to literally force your proteins through using electricity, using voltage, to sort them through to size. So you're forcing them through like literally a gel, and they're going to separate based on size. The big ones are going to stay up near the top, and the small ones are going to go through quicker. So it's just like getting sand and stones and just pouring them through. So yeah, I'm going to play you this video, which shows you what that's like. Unfortunately, they're saying, oh, you're here. So that's your apparatus for passing the the bulk through, and he's making up the gel now. It's a 12% charged gel, and you literally have to clean all the plates, put them together, and pour them in between the two plates, and you're basically just setting a gel, and that's all he's doing, but it takes forever. And he's using just literally a piece of meat for his protein sample, and then he's putting a dye in. I don't know if you can see that. So you put a dye in just so that you can track the progress of your protein through your gel. So then we're applying negative to positive charge, and the proteins are running through because they're negatively charged. They're running towards the positive point. He's put a marker there on the right hand side. So these are known proteins of a certain size, so you can kind of determine where your proteins are going to come. So that's the gel you get at the end. So that's sort of exactly what it looks like, significantly spelled. So I'm just going to show you exactly what it looks like when you're running the gel. Now this is over about an hour. So you put your proteins in with your loading marker here, your loading dye. That's showing you that it's blue. They're moving through. The dye is going to move faster than the proteins because it's just a dye. And so it's going to go through to the bottom. That will show you when you need to take off, stop applying the current. So your proteins are literally, you can't see them because they're not colored, but they're just sorting out based on size. So what would happen if you can see over here, you can't see them on this. So that's actually what it would look like. If I took a dye and stained all the proteins that you have in your gel, this is sort of what you would see. So you can see that you've got just bands of protein everywhere, because there's a lot of proteins in our sample. And that sort of is, that dye is going to dye everything that has amino acids in, so it's going to dye every protein. So that's really great. I've got all my proteins on a gel now, and I can sort them based on size, but I still can't exactly pick anything out from there. I can't make any sort of meaningful information. We've got all our proteins on a gel and they're all stained. So what? That's when we need to do Western blots. So we need to get the proteins out of the gel. They're not very useful in the gel. Onto a sort of a substance we can use. So Western blotting is taking the proteins from that gel, where they're sort of like size, and putting them onto a paper-like substance. So we use a membrane of PVDF or nitrocellulose. So we do that again using electricity because our proteins are already charged. So basically what we've done is run the proteins down through a gel. Now we're going to actually run them out of the gel onto a piece of paper using the same principle. So that involves literally making a sandwich like this. So she's got her gel there, I think you can see it, and she's going to take out a piece of her nitrocellulose paper, and she's going to put that on top of the gel, and then what you actually need to be really careful is you don't get any bubbles and stuff, because then you're going to fold over that sandwich, and you're going to immerse it into a bath of sort of ionic solution, buffer, and you're going to pass the current through it the other way and take the proteins out of the gel and put them onto the paper. And again, this takes about an hour. And I'm showing it to you in 2 minutes. Oops, stop doing that. Okay, so you're going to put about 400 milliamps through that for about an hour, and then you're going to get all your proteins from your, hopefully, if it works, which it doesn't always, you're going to get all your proteins from your gel onto your nitrocellulose. Okay, so we got cells, we broke them up, I made a ridiculous contraption to sort them by size, we put them on a piece of paper. Why did I go to all that much trouble? I've now got a piece of paper with a load of proteins on it, and I still don't know which proteins are on it where. Well, we all do that so we can do the most important part, the most interesting part, which is chemiluminescence. So I've showed basically this picture here of a scientist with a big beaker of luminol, which is sort of what we're doing. We need to make an antibody directed towards our target protein. So we have an idea, say we want to look at a protein and proteins on the outside of HIV. We need to design an antibody that is going to recognize just the protein we're interested in. So how do we do that? And it's basically just like the principle of immunization. We allow our protein to kind of be introduced to an animal, kind of like immunization, and they're going to recognize that as an antigen. So they're going to start producing antibodies towards the antigen, which is our protein, and then we're actually going to try and purify those and cultivate them. So you basically want to allow, you're going to take the antibodies out of the serum of the animal and then purify them down, and you get like a little vial of antibody eventually, and that takes months. We literally just buy ours. I wouldn't have time to go and do this. This is would be a whole PhD project in itself, would be designing these sort of antibodies. So then what you need to do is you take a piece of paper, you put your antibodies on it, and you leave it alone for a while for your antibodies to stick to your protein. You allow the antibody to stick, and then you're literally just going to wash it off with water, salt, and detergent again. And then you take your antibody-covered membrane, and that actually has a fluorescent tag on it. So what you then do is activate the fluorescent tag using hydrogen peroxide, and you're going to put it under an extremely expensive, extremely finicky camera, and you're going to try to detect the amount of light coming off your blot. So the light should only be detected where the antibody is going to be bound, and that literally is what it will look like. So you can see, I was saying we sorted our protein by size when we did our gel, so that's why we get all these different sizes, because I said we ran a marker alongside. So these are all the different sizes. So this protein here is between 50 and 37. Okay. Then we took our antibody, we put it on top, and it binded just to our protein, And then we have a fluorescent label on it, which we then activate, and then it will actually release light, and then we capture that with a camera, just like the same principle of any photography. So that's sort of what you get. You spend 2 days at least, maybe 3, to just get this one image, which would be, this would be like sample 1 to 10, it could be different conditions. You can see from sample 6 to 10. This protein is actually actin, it's what makes up your cytoskeleton, the outside of your cells. And this is basically a blot that I would have done. So I work on a prion protein, and it's the protein involved in BSE, mad cow disease, and the infectious form of the prion protein causes mad cow disease or scrapie in sheep. We all have this protein, but in the non-infectious form in our bodies. In fact, almost every mammal does. So what I would normally do is I have infected cell lines, immortal cell lines, and non-infected immortal cell lines. So what I've done is I've ran my gel, put all my proteins on it, and then I've probed for just the prion protein. So you can see in this line here, this is my non-infected cells, and you can just see the protein comes up. What I then do is, because the infected form of the prion is actually really, really hard, it doesn't digest, it's resistant to heat, it's resistant to detergent, it's actually really resistant in general. To show that my cells don't have any prion infectivity in them, I digest them with the protease, and you can see there's nothing there. So once I digest my sample up, there's no prion in there, nothing coming up with the antibody, which means there's no infectivity. Then if I look at my infected cells, that's my uninfected, that's my digest. These are my scrapie infected ones. So I haven't digested that length, that's just my normal sample, that's what it looks like. It actually looks, you can see it's darker, but it's almost the exact same. It's darker because there's more protein on it. But there's no real difference between my uninfected until I digest it again. So when I digest that with PK, proteinase K, you can see there's still protein left, and that protein is all my infected prion, because it's just completely resistant to basically everything. So ultimately, I guess what I kind of wanted to say was actual science, a lot of physical work. The videos I've shown you showed like a lot of like putting glass plates together, pouring gels, and like putting sandwiches together. It's actually a very little brain work during the day on some levels. It's like literally taking sponges, wetting them, putting filter paper on top of it, putting a gel on it. Like it's a lot of actual physical, somewhat boring work, I guess. And I guess it's just a lot of patience, resolve, and persistence. Everything else is just being fancy, and the results take a really long time. Some days can be really repetitive, and other days incredibly exciting when you get really good results. And everything I showed you, from the gel is neurotoxic, to the methanol I use in my buffers, which is also neurotoxic. gel is also cancerous. Like everything I work with like gloves and massive precaution. Obviously, immortal cell lines aren't the best either to be letting out into in general. I don't want to let prion proteins out, so there's like loads of waste that I have to like bleach and things like that, and then put the bleach all over your pants and wet your pants, just so I never wear any nice clothes. So yeah, everything is really toxic and dangerous in the lab too. So I guess immortal cell lines are like looking after a pet. Protein lysis is just smashing with detergent. Separation by SDS page is just forcing proteins through a gel mix. Immunoblotting is just putting them on paper. And immunogenesis is sticky glowing net mixture. That's it. Thank you. What is any other question?
It's probably a stupid question, but I'd like to hear the stupid answer anyway. Is there a way of automating this? Like, I assume it's very expensive to do, but are there systems that automate this kind of thing or speed it up?
There are systems that will grow your immortal cell lines. So like there's automated tissue culture, which would be like all like then you have to have a camera that watches to show, and a pH probe as well that will tell you when the cells are grown, like when they definitely confluent. So basically, when I say confluent, I mean the cells have grown so much there's no more room in the dish, basically, and that's when you have to separate them, put one half in one dish, one half in the other. And yeah, there's like, and they have to have like sterile tips so that they'll suck off the sort of wasted media and then like remove them. Yeah, that's really expensive, like insanely. Nobel Prize money would just about buy one of those sets. And to be honest, it's not really practical because they work on like, so these little dishes like this, and they'll have, if you can imagine, just 96 different little wells, little petri dishes within a dish like that. That's sort of what they work on that scale with a machine like that. I use dishes that are about this big, and I would use maybe one of these dishes to do one sample. And one sample isn't going to get any paper, so I have to do like hundreds of samples. So it's not really practical to have it automated. There's so many steps. Like you see, you have to run the gel, and like I just can't see how you would automate the entire thing. There are ways to automate, and also different ways to do what I've shown you. Like there's a million different setups which will essentially just pass protein through a gel. There's different ways, different widths of glass, and like all this sort of stuff. And then with the transfer as well, like there's different ways to pass electricity through the gel. Some people even use suction to suck the proteins out onto the paper and things like that. Some people will look at some people try to look at the proteins as well in the cell culture. So instead of even breaking open the cells, again, that's all different expenses and different techniques. But this is sort of the main one that would be used for proteins.
So the answer is possibly automatable, but way too much effort.
Way too much effort. It's way cheaper to just employ a PhD student for 4 years than and actually 10 PhD students for 4 years, yeah.
So you're saying there's a lot of like physical work in it.
Yeah.
What's the role of like compartmentalization in the lab, where like you kind of a tech do this and then
why have a tech do anything? You don't have a PhD student do it all. Is that what you're sort of asking? Like one person who would always run those gels?
Yeah. Well, yeah. I mean, how's that kind of structured? I mean, you see the movies where you have the lab boss who's in his office, and then everyone's kind of running around in lab coats, and he's just barking orders at him. Is it like that?
Yes. Exactly like that. Yeah, my supervisor once told me PhD students are like just like little lab robots. Actually, if she could invent lab robots, she would just fire all of us, which I thought was really nice. But really, like, yes, for me, doing a PhD is very like, well, it should be that you have a lot of sort of direction with the project and things like that, so you want to actually do these experiments. But there is a lot of just like, you have to do it that way. I mean, you have to just go and make the gel yourself, and then you can buy the gels pre-done, again, more expense. You can then, buy your protein samples in as well, but at the end of the day, that costs so much money, and you only have so much money on a grant. The realistic practicality is you don't have someone helping you, you literally just do everything from A to B. But that's sort of rewarding as well, because I know that from from the cells down to the very end image I get. I've done everything. So,
Well, sure, eventually you'll have to be able to tell the lab robots how to do their job, so you need to know how to do it.
yeah, exactly. So, I tell them that I did it with less money and less.
Back in my day,
Exactly.
it
Yeah.
cost half as much.
Yeah. So, but yeah, a PhD definitely is about sort of doing everything.
Right.
Bear in mind this question might come from me not understanding things properly, but if this is a process of sort of separating and highlighting the proteins that you use, so what's the end game like? What do you do sort of after this step, or what's examples of what's done after this step?
Okay, so like why would I really want to go through all this? So,
Exactly.
Yeah. So no, that makes perfect sense. So for example, I work on a crime lab. So we work on cells that are infected with scrapie, the sheet form of the prion protein. And one of the guy's projects was on he thought he was working on cyclodextrins, which are basically the chemical that's in Febreze. And he was finding that when you put certain types of these chemicals, like there were different chemical bonds and stuff like that, different sort of modifications to the chemical, but certain ones were effective in clearing the scrapie. So what you would do is, he would have a range of concentrations of the drug he was putting on. He would put them on the cells for say 3 weeks, so he'd have one where he'd put on like 100%, 180, 160, 120, and he would see what was the minimum concentration over 3 weeks to actually get rid of all the infectivity. So then he would get all the samples like I've just shown you, do the entire process that I've just shown you, and then he would get to this. And he would see when he, for example, if he added 100% of his stuff, that there would be no scrapie left when he did his digestion. And then he would show that, you know, if he added none, he would still have the scrapie left. And what you'll do is you'll actually get data from this by, you literally draw a box, in a program you draw a box around the area that's, you know, being highlighted, that's in black. And you show that this area is about 50% as strong as this area, therefore there's 50% more protein there. So that's what you eventually show as a graph that will say 50%, which is really depressing. And then you show them the picture and say like, see, it's 50%. And that's genuinely what you would do. So that's the sort of example of what you would do. You treat your different cell lines with certain things, or you put them in different conditions. You can like knock out proteins and see what the effect is. That's like more stuff. It's like, I was trying to keep it somewhat like basic, but yeah, you can upregulate proteins, downregulate proteins, and then see other effects. So you would basically show that your protein is upregulated, and then take that block, because if you can imagine, that piece of paper has all your proteins on it. So then you would try wash off your fluorescent antibody, and then you would put on another antibody directed towards a different protein, and show when I upregulate this protein, it actually affects another one. And you'd show that this one's higher, and then also this other one's higher, or this one's a bit lower, or something like that. So that's really sort of what you're trying to do.
Yeah.
And it's really arduous, and like the stuff that Alyssa and Claire do generates like a lot more data a lot quicker, and I think it's a lot more. I definitely don't want to do this when I finish my PhD, basically. But yeah, this is the only real way. So I think protein science has been like this for about 30 years, while other like DNA and stuff has taken these huge leaps in sort of what we can do. Protein science has stayed a lot the same in terms of like a Western blot done now and 30 years ago is the exact same. It's the same process. That's it. Is that right?